It is possible to create a life through a scientific experiment. What are the consequences of emulating God? The most complete testimony of these issues was totally the sheep. In the early 19th, the cloning of living beings from DNA has a recurring theme that exists in the field of science. The successful movie Jurassic Park helped popularize it and inventing fantasy. This movie shows how animals went extinct millions of years like dinosaurs. 100 million years ago, there were mosquitoes, just like today, and just like today, they fed on the blood of animals, even dinosaurs. Sometimes after beating a dinosaur on the branch of a tree and get stuck in the sand after a long time, the tree sand would get hard and become fossilized just like dinosaur bone, preserving the mosquito inside. This fossilized tree sand, which is called amber, waited for millions of years with the mosquito inside until Jurassic Park scientific came along. Using sophisticated techniques, they extract the preserved blood from the mosquito and bingo, DNA. Now that's where the genetics take over. Gene machines, supercomputers, and gene sequencers break down the stand in minutes, and virtual reality displays show the genetics, the gaps in the DNA sequence. They use the complete DNA from a front to fill in the holes and complete the cut, and that's how they made a baby dinosaur. So, in order to make an attractive and profitable park, it would be necessary to have dinosaurs. Approaching this movie to the real life, July 5 was born only the first successful cloning of a mammal. In the Russian Institute of Edinburgh, Scotland, in 1996, Dr. Ian Wilmot used this technique to clone a 6-year-old Findoster white sheep. In the process, an egg without DNA and without a nucleus was first knit. For this, he removed the nucleus of an egg from a black sheep using a very fine cold needle. Then, instead of fertilizing the egg with a sperm, he had to introduce a nucleus with DNA from a somatic cell that is a cell differentiated from any tissue of the sheep. That is what decided to clone, because all these cells have the same information. The organism turned out to have the same instruction plan as the original sheep, and it should develop in a similar way. Finally, the embryo was implanted in the uterus from a female of the clone species from gestation and birth. This sperm was made to obtain sheep with pharmacological properties and beneficial components, and ended up demonstrating that a living being can be cloned from a differentiated adult cells. This generates promising exploitation in the medical, livestock field. In science, it can be a source of obtaining of 100% countable replacement originals for a transplant in a sick person, or produce transgenic animals with artificial characteristics such as resistance to viruses and bacteria. And a lot of savings will be made in medicine. So these are some of the thousands of benefits that the development of cloning could obtain only in humans, but also in other organisms that it can benefit us. The cloning of animals is really a reality. 26 species, but it will be not long before we will be able to make clones of people. But even if we can't really create them, we want a society with replicas of human beings, to have a copy as not feel alone, or simply because we want.